Thank you so much, Nico, for the great explanation. So today I'm going to talk about that's almost we tried to submit the data, the whole entire data. We challenge a little bit higher journal than usual. So this is a work of the collaborative work with North Carolina University, State University, lead Roberts, and then also Farzana Nazarene. She used to be in the UH Manoa in the math department. She now moved to the University of Tennessee, Knoxville. Okay, so we use the cavefish and surface fish, the Mexican tetra, this fish in our animal facility. Let's see. Well, it's always happened to my lecture too. Yeah. So the first I didn't start gut microbiota with the C-MΑIKI team or the Margaret McFall. We already started gut microbiota in 2015 with a collaboration with Robert. Our question is the gut microbiota. We have the cavefish and surface fish. It's a super distinct environment, and how the gut microbiota are going to play a role in the distinct environment. Okay, so the gut microbiota, I don't have to tell so much in this group, but the gut host genetics and the fish genetics is going to influence for the gut microbiota the profile, as well as the diet, the fish eating also going to change the gut microbiota composition, of course. Then the maybe I'm too far. Then the gut microbiota also going to influence the host genetics to change the gene expression in the gut, or maybe in the brain too. Then and is also going to influence to the gut morphology, as well as the gut morphology is going to change the gut microbiota composition as well. So the genetics is more developmental processes for changing the gut morphology, right? All in all, this is going to put it into how the host growth, host health condition, and the host homeostasis, how it's changed. And also giving the, if it's a good relationship, it's going to give the healthy condition. Okay, so maybe I'm too far. So I'm right. See. So we are interested in for this network and how it's going to relate it to each other for the gut microbiota and the host nutrient, gut microbiota, and the host growth. Today's theme is host growth. Okay, particularly for the host colonization into the different environment, new environment. Just thinking about the evolutionary path, the animal, I'm paying attention to the animal too much, but for the plant as well, the massive extinction happen when the environment change, then that new lineage of the animals, new species came up after such kind of environmental shift, right? We always think about the abiotic factor like temperature, oxygen concentration, but what I was keep thinking about is probably the diet, also the microbiome in the novel environment could influence or facilitate animal evolution. That is I keep thinking about these days. So the diet side, nutrient and gut microbiota could promote to adapt into the new environment. So we are working on this the idea and then try to look at how this gut and animal relationship evolution. That is a big theme for us. So our fish system, they originate in the northeast side of the Mexico. there's a limestone mountain running north to south, and this limestone mountain have a multiple cave. There's a study cave which contains cavefish over there. And then many of the cave, this is about 60 miles away, 100 kilometers away, and 100 kilometers away. This cave fish is the migration rate is very, very low in the population genetics, like less than one fish is going over in each cave. So we think this is independent and independently evolved these different caves. So convergent evolution. Okay, so it's convergent evolution of this phenotype, eyeless, pigmentless phenotype. And then its time scale is about 20 to 200,000 years. Given surface fish is still interfertile, which is a very strong advantage we are using this model system. Convergent evolution model evolved under the nutrient poor and dark environment. And as model animals, they can be adult in 6 to 7 months. That is kind of slower as a model organism, but we can still do the embryology, microsurgery, as well as CRISPR-Cas9, okay, for the host site. And then their evolutionary, the phylogenetic relationship is very well characterized. We're gonna talk about this surface population, which is it's called a new lineage, but they are abundant in this entire area here. Morino cavefish, this one, is the closest cavefish, phylogenetically closest cavefish. Okay. Then that is colonizing this here, and then these two, Tinaha and Pachon and Tinaha, they are more diverged from the current surface fish population. Okay, I think the recorded video is moving, so don't worry about it. So the research question today that I'm going to talk about is how did cavefish overcome the growth of the lean environment? And then next question is how do adult cavefish incorporate its starvation resistance with gut microbiota? Okay, I'm going to spend about 2/3 of the time to this talk here, and then just touch on this bottom side. Many people working in this part, adult cavefish side in our community. Okay, so the fish, surface fish eating this, they are carnivore. They are omnivore. They can eat the carnivores, the animals, as well as the plant they eat in the environment. The cavefish, they usually eat bud guano, which is the bud colony nesting on the top of the ceiling. They drop their poops, and then cavefish eat. As well as occasionally they have the cave-adapted the arthropod. They seem to eat, but they are not aiming too much to this cave-adapted arthropod. Okay, I can tell the reason is, but yeah, just for your note. So first we take a look at the growth, how it's going to relate it to the different diet. Okay, so we're gonna do the different diet composition. We have a different population, surface fish and cavefish, Pachon and Morino. Morino is just a reminder, it's closer to the surface fish than Pachon. Pachon is a further derived one. And we have F1 hybrid of each surface fish and Pachon, surface fish and Morino. That's population. And can take a look at the genetics. Environmental factor is we gave the nutrient-rich live brine shrimp or live brine shrimp egg that is this caloric amount. And then we thought that we also gave the like a plant-based or the spirulina is not a plant actually, but it's the chlorophyll containing diet that is slightly leaner, and then but guano that is like the natural food we think, and then we do not know about the caloric amount. Nobody can measure it yet. It's super lean. Then we did the 5 day. We raised up until 5 days old. This species start eating at the 5 days old,
And then up until the 1 month old, we raised with live brine shrimp. We tested this diet from the very beginning, but they die because they cannot digest well and they cannot survive. Then 1 month of the experimental, the diet experiment, and then we got do the gut microbiota study. Okay, so the fish, after the 1 month of the raise, this is again this is just a 2 months old fish, 2 to 3 months old fish. They are still under the development, rapidly developing. And then brine shrimp, spirulina diet, and the bad guano. This is surface fish, and this is each individual fish. So this is one example, another example in the brine shrimp diet, spirulina and the bad guano diet. They are really suffer the bad guano diet. They are so super small. Gut size is also small. What we surprise is spirulina giving the much better growth in the surface fish, Molino, and then Pachon. Again, we didn't know that they are more herbivore type of the preference or the adaptation in the gut, but the point is the spirulina is a leaner diet, and the brine shrimp is much more commonly accepted diet, which is very easy to digest, and then the common fish diet, larva diet. But for our case, the spirulina make the fish grow better. All right, so this is a gut structure. This is drawn by the very talented undergrad student, Crystal Valdez. She graduated in 2018 already. The stomach, pyroxica. Pyroxica is a kind of apex-like structure, and the human usually have the boundary between midgut and hindgut, but the fish have this pyroxica. On between the stomach and midgut, the role is kind of similar to the human apex. They have it is a sac, so it can have a much more gut microbiome here. And then there's an immune cell attached on here. The immune cell is educated on this pyroxica, so therefore they have a good adapted immune system from there. Okay, so this midgut and hindgut. We're going to take a look at the gut length of the midgut and the hindgut here. Pyroxica structure itself is, we also counted, but it's not so much interesting because it's more defined by genetics. So the body growth, how it is grow. Here's the surface fish. These 3 bar is surface fish. These 3 bar is Pachon cavefish, surface fish, and the Molino cavefish. This 3 bar is exactly the same as here, just a difference to compare with surface fish in Molino. And this is F1 crossing with a Molino cavefish. F1 crossing with a Pachon cavefish here. Okay. Then this blue star is comparing with brine shrimp diet against the spirulina or bad guano. So if you see the star, that is the difference between the the brine shrimp with bad guano, or brine shrimp with spirulina. Okay, all right. And then in general, the cavefish grow faster than surface fish. Okay, so surface fish is smaller than the Pachon, or smaller than the Molino cavefish. And then of course, the bad guano is less efficient to grow. There's no star here. It's not statistically significant, but you can see the trend here. Okay, again, you can see the spirulina makes the cavefish grow faster. Surface fish also a little bit, but not significantly higher. Molino is not significantly higher, but you can see it's exceed. All right. Here you may not see so many dots in here. That is not the error. It's actually the Molino fish died with bad guano. They just cannot tolerate with bad guano. So I think there's 2 points still. Just 2 individuals survive out of the 12 individuals we studied. Okay. Anyway, that is very detailed. But point is, body size larger in the cavefish, and then body become larger in the spirulina diet, which is the leaner diet than the brine shrimp diet, which is a big surprise for us. So that is driving us to maybe do something doing on the gut microbiota to make tolerate or taking more energy from the spirulina. This is gut size. The take-home message is also kind of similar what I showed in the fish length. The standardized total gut length is standardized with the fish length. And then you can see the guano is not, but guano is not good for the growing the gut system, but spirulina still has some effect for the growing better. Okay. So this is a summary of some diet effect and genetic effect. Maybe the body size have a genetic effect, but diet effect also there. Paroxysika almost no diet effect, but the genetic effect is higher. So this is just qualitative counting in here. All right, then we are interested in the gut microbiota. So far, do you have any question? Are you okay to move forward? Okay. So the gut microbiota, just going to the relative abundance plot. First I'm really disappointed. We couldn't see so much of the big difference between the population. There may be a difference in the diet. So this is gut. G is but guano is some difference in this F1, but not clear pattern. And of course, also this pattern is quite different from the diet microbiome, the tank microbiome. So maybe it is a good sign that is the fish microbiome is selective. They try to attach on there. Okay. Then so gut microbiota branching are similar between the different population. Yeah, gut microbiota is different from the diet tank water sample, right? Alpha diversity, not so much significant. Again, we disappointed that it's not so much interesting ongoing. Then beta diversity either. So somehow this is the same plot between left and right, and then some change in the diet, but not so significantly strong. All right, so we stuck for a while, this part. Okay, like I said, we started this project 2015, and then we still we didn't publish this data because we couldn't pull out something important from there. Diet shows slightly better separation. Because we believe something ongoing by looking at the spirulina, they grow better. We could detect some signal from the gut microbiota, but this popular method didn't show us anything. Okay, so we step in. So this is I think if you are in the corporate team, I presented it multiple times. This pipeline we developed with the Farzana, who is in the Tennessee now. The challenge is to take a look at for data structure. We usually use a PCOA, PCA, or NMDS. The challenge is that we going to lose the information in when we are doing the dimensional reduction, or skewed profile if we use the UMAP or TSNE. Okay, so that's the distance matrix is not proper on that case. Also, if we want to see the microbe which is associated with, let's say, growth, we need to. The best way is a regression. We don't do the correlation analysis because correlation losing many information too. I can tell why it is. So and then the current modern the preferred method is random forest to identify the best the strong effect microbiome out of that massive data set. But if you only have a very small data set, the good productive split couldn't made in the random forest. So the Farzana and our lab member Iwashita, they try to insert this new methodological data analysis method in the sparse modeling. I'm going to talk about this one. First is the topological mapping, or it's called topological data analysis. What you want to do is try to simplify the data, like a manifold structure of the data, into a very simplified version of this one, topological map. You have originally have this type of data, right? It's maybe the multi-dimensional. If you have the 600 of the microbe or the 1,000 of the microbe in the human case, then you have the 1,000 axis degree of the data set. 1,000 axis, you plotted this data. Each plot is each individual fish. First, you're going to project into the one-dimensional something measurement. Many people also in the TDA field, people in the math, they use the first axis, well, principal component 1. Our case, the Farzana is very creative. She suggested to use the growth, like fish growth axis, 1D direction, 1D data set. You can project that fish growth to your data to there. Then you're going to make a bin. So that each bin, according to that the scale. Our case is growth. Many people using PC1, the score and the bin, and then each bin you're going to apply the cluster algorithm. Okay, so this red bin you're going to apply the cluster algorithm, and it's the cluster algorithm and just say that there's only one cluster, then you're going to give the one cluster. If it says a 2 cluster, like this bin level. There's 2 cluster, it's the cluster algorithm tells, then you can give the 2 clusters there. Okay. And the cluster algorithm, you can use the PCA or the TSNP or UMAP, whatever you prefer, the cluster algorithm, you can use it. So you can make a multiple clusters there. And then the important point is that you intentionally have the overlap of the bin here. Then this overlap inform the algorithm to make, okay, so this cluster and this cluster is close each other. This cluster and cluster is not close each other. So that if it's close enough, then the link is provided for this. Okay, so its topology is preserved, and then no data loss at all. And then you can see the 2-dimensional structure in this way. Okay, whatever that just mentioned it. And then in the very beginning, we didn't see the big advantage to simplify in this way, but I'm going to talk about we found the best use of this method. So if we make this topological mapping on the same data set. This is the NMDS result in the former slide. It's very mixture of the surface fish and cavefish in different population. And this is the diet side. So you can see the different combination. Each pie chart representing the individual fish. The larger pie chart representing more fish. A smaller pie chart means a less number of the fish in the cluster, and this pie chart split into the like maybe a third of this pie chart is made out of the fish individual, which is fed with guano, right? And then a third is spirulina, and then rest is branching fed individual. So this is representing how this structure, this cluster is made out of the individuals. All right. Okay, so we have this one. Okay, so before we move forward to telling about the advantage of this cluster, I'm going to talk about this is I already mentioned. Okay, the second step part, that is a sparse model. So how you're going to regress against to the 1D data set, like for our case, the ASV against the growth. All right. So the regression analysis always have a challenge. You have a small sample size. Maybe you have a 50 individual fish, And then you have like 1,000 of the ASV. That's the things towards regress against there. Each coefficient of the 1,000 of the 1,000 variable is over like it's overestimated, or it's not mathematically it's not going to work for predicting that the coefficient. And then this is we just applied the random forest, and they pick up about 100 of the coefficient out of this the ASV, and that doesn't make sense because we only have 50 fish, right? Does it make sense? Okay, so sparse modeling. This part is normal regression model. So the Y is the growth for our case. You have weight with ASV1 count, weight of the ASV2 count. You can do it. Is a normal multi-regression. The sparse modeling is adding the penalty here. That is going to evaluate each variable if that is be included or not included, just very objective way to drop or include these ASV. Therefore, we can overcome this issue of the small sample size with a very large number of the variable. Okay, so this is a flow chart for the ASV data set. You can have the row is individual fish or sample ASV here. Then you need to first normalize with we use a geometric mean pairwise I forgot, but the normalize of the each count. Then make a topological map by using the ASV, right? The cluster here. This is the population, sorry, the standard length 2SD to the negative 2 SD. Then this map is also you can use to color with the diet to whatever. Then what you can do for this is the strongest point for our method. You can see, right? So this is the longer fish. This is also longer fish, the larger fish, and this is smaller fish here. Okay, smaller fish in this cluster. Important thing is if this is a different cluster, which means their gut microbiota is different, right? These are quite different gut microbiota, right? Even they have a longer, the larger body size. And then the challenge is we usually have a challenge that is this type of noisy data. If you just try to regress the ASV against the growth score, you cannot find any variable that is going to associated with or important for the growth associated with the growth. But if you splitting out, you can split into the big one to the small one, big one to the small one, or you can trace other path to small one, and then apply that sparse model or regression model to then you can identify some ASV is strongly related with growth. In particular for this cluster, subsampling, subgroup, or this subgroup. We frequently see that some subgroup couldn't give any of the ASV associated with the growth, but some other is associated with growth. So this is the way we try to achieve to eliminate the noise out of the noisy data. Why this is I mean the population is so different. This data itself is from the surface fish. Okay, the only surface fish. Surface population, they are polygenic. They are not the inbred line. Maybe that the genetics is going to contribute for this distinct pattern, or maybe even the very beginning when they start eating, they picking up different type of the microorganism in the very beginning. So therefore, that is colonized in the area. That is the factor that's going to swing their gut microbiota this way or that way. any story you can think about, but it is make us difficult to analyze. But this way, we can clean up the result. This is another little bit detail, but how you apply the sparse modeling, we do the sparse modeling, the very fine tune. It's too much detail, but if the sparse modeling contains the lasso regression as well as the ridge regression. These two regression is penalizing that's the variable. But the majority of the data set, we do not know how much we have to weigh on the L1, like lasso regression versus ridge regression. So you have always a balance between the ridge and lasso. So you're going to search from the L1 rate means lasso is 0.1 and ridge is 0.9 weight towards completely lasso regression. You're going to simulate multiple times because each result, there's some the stochastic condition. So you have the different answer in the 10 runs, but which the different answer means the different ASV selected in each run. But if we see the more than 8 times that the ASV came up in this test trial, we selected that that ASV as a robust one, and then so that is a robust one. Then we also try to see the single regression between one ASV versus growth, and then see whether that is actually giving the high score of the R square. Then followed by the univariate justification on here. Sometime the N means the many of the fish individual have the 0 score of the ASV. So we just exclude that that's the ASV if they have less than 10 spot to make this regression. If it is more than 10 or more, if we see that this plot, non-zero plot, then we recover that ASV. And lastly, we try to recover the ASV, which is highly correlated with the selected ASV. Okay, because we're thinking about that quantum the effect of the microbiome, or these highly correlated microbes could help that the microbe which is we pull out from this data set. So that's correlated more than 0.95. The count variable is correlated with more 0.95. Then we pull that ASV from our data set, recover the ASV from our data set. So we carefully do it in that way. Then this is the full view of how which microbes is pulled out from this growth, the fish length, the regression. Okay, so the surface fish in here, Morino cavefish and Pachon cavefish now here. And then you could see some of the microbe is kind of shared. So this Plesionomas is it have this plus or minus means positively associated or negatively positive correlation, negative correlation, or positively associated, negatively associated, right? So this one, if this one is a larger number, the surface fish doesn't grow well. Okay, so this one is shared with the Pachon cavefish. But interesting things is many of the other genera like Rhinorea and other one have the direction change. So this is positively correlated in the surface fish. Like if this is more, the surface fish grow more, but the negative in the cavefish. All right. The color, the same color is the same genera. Okay. Then And then this one is newly emerged. Enterobacterus and the Enterobacteriaceae is newly emerged in the cavefish population associated with the fish growth. Okay, so well, again, this is our computation result. So we are not sure if this is actually the real or not. Okay, so we try to disturb this gut microbiota with the antibiotics, and the question is if the same genera is pulled out in that antibiotics treated one, then probably this is like we can see this the genuine result, or maybe this is real. That is the idea. So we treated the gut the antibiotics. So we fed the gut antibiotics to the fish. It doesn't change the standard length, right? The body depth. So it doesn't affect for this fish at all. And then gut length slightly reduced after the antibiotics treatment. I mean, comparing the control, it's reduced gut length. But again, I can tell this is mild because fish are growing well. Okay. So when we look at, so this profile didn't change so much, like alpha diversity and beta diversity doesn't change in the antibiotics treated. So this is very gentle and mild condition of the antibiotics treatment, right? Then take a look at for this, to the right side of the antibiotics treatment, treated a surface fish in patron cavefish. Left side is just as shown in the on the former slide. It's only and this Legionella is consistently appear as a negative value here. Okay, so we could successfully recover the same the genera. I mean, it's already regressed very reduced pretty much because of the antibiotics treatment, but we could still see if that genera survive out of the antibiotics challenge, then we can detect the same one. Okay, we could detect the same genera. Okay, so we couldn't identify the species level. All right, so it seems okay. So our pipeline seems okay to work, but the point is, the important point is we are not so much satisfied by just pointing out the microbes, right? Microbe is okay. We knew but maybe this is just for our fish, right? It's not so much generality we have. This is only for the Astyanax mexicanus. This is happen in the past. This is helping for their growth, but so what, right? Or maybe that is only specific for our fish evolution. So what we do is we use a PyClass tool, which is going to estimate the pathway, enrich the pathway in the gut. It is originally built up with the human gut microbe project, and then and also pathogen based one. But it is enriched. It is every year. And then it also contains environmental sample too these days. Again, but it's still more biased towards the human side, not if it's unknown. Let's say that is if your animal is very, very distinct, and then this estimation may be not good for the estimating the enriched pathway. But anyway, we tested it. So okay, so what we did is this is just ASV, the same as the last one, and then PyClass 2 is going to estimate the enrichment of each pathway. This is now the pathway name here instead of the ASV name. Then do the same things for the combining with node selection with the topological mapping. So here, and then continue to top here. Then we do that's sparse modeling to identify the pathway. Okay, so all right. So this is growth, and then the positive is positively associated, negative is negatively associated, and this is a very tiny letter here. But this is color coded. The orange orange color code is a Pachon cavefish. Morino is a pink. Okay, so the positively associated one, particularly here, these are all Pachon fish, right? That's a big surprise. And then Morino is in here. So there's some negatively associated microbe is there. So just magnifying there. And this majority is some Morino, the pink color here, and the majority is the orange color Pachon cavefish. And then many of them is acetylene degradation, starch biosynthesis, and nitrate reduction, blah blah blah. But it seems like many the glucose related or starch related process, glycogen related processes seems to have a positive association with the growth in the Pachon cavefish. That is the further derived animals. Carbohydrate metabolism. The negative side is more microorganism activity related things that is maybe pathogen. We don't know that is associated. Then the yellow highlighted the part. This yellow highlighted one. Fortunately, we have an inhibitor for this yellow highlighted pathway. That is quercetin. Quercetin is a human drug as well. The plant extracted the chemical, which is used for the obesity patient to reduce the blood glucose level. It also known inhibitor for this process in the glycogen degradation. I forgot what it said. And starch degradation, the quercetin here. So the yellow highlighted part in the former slide is the quercetin target. We treated the quercetin in our fish. So the control and quercetin, we raise the fish, so body standard length, 0 week. This is started from the 3 months old, so it's still on the growing process. So the 1 month of the treatment. Okay, the gray is a control diet, and then the red is quercetin containing diet. The Pachon is, so this is bad guano, so the growth is very slow. Bad guano is very slow growing. Control Pachon fish growing with a star. It's the the quercetin treated one is non-significant. Morino is both growing well. Okay, so this is a standard length, the fish that size. Body weight is also the quercetin kind of suppress the growth there. Okay, we also did for the spirulina diet. So this is a spirulina diet for the 1 month. For the from the let's see. From the 0 week to the 5 weeks, this is a 2 months old, 3 to 2 months old fish treated 1 month. So they are growing. You can see the 4 weeks old, the control diet is kind of exceeding the growth. Spirulina is more nutrient rich, so they are growing much better. The Morino doesn't respond well. The quercetin doesn't interrupt their growth so much. And then this effect is kind of, you cannot see in the, if you use in the young adult stage, they are still growing slightly, and this 5 weeks, you can't see so much of the growth help in the larger or almost adult fish, right? Okay, so the conclusion from the former slide, this slide is showing the surprising result that is the Pachon cavefish evolves to receive some nutrient from the gut microbiota, which can be suppressed by the quercetin, to make a rapid growth here. That is reduced by quercetin. They evolved to receive the new benefit from microbe in the cavefish, which is not happen in other cavefish, Morino cavefish. The distinct pathway. So that starch, glucose, glycogen processing containing the enriched the microbe helping the Pachon fish growth. So summary, Pachon cavefish grow better in the spirulina diet And then the analysis pipeline, I just show you how to confront with the noisy data. Cavefish evolved the new association with gut microbiota for the growth, gut length, and gut width. Pycrust still seems to okay because quercetin work, right? But even this noisy data, Pycrust may make a good prediction, and then we can knock off the one serial pathway by using the drug. Pattern cavefish evolved faster growth by the new association with the gut microbiota. That is going to probably help for the lean environment. They need to grow even the lower nutrient input. All right. Okay, so this is the first section done. Rest is probably quarter, not the third. Do you have any question for up until here? Okay, then so the many people in the cavefish field, they are asking for how the starvation resistance associated with the gut microbiota. So this is a full adult. You can see this is more than like a 4 or 5 centimeter, like 2 inches or 3 inches length, right? Then this is control and the fasted fish, female and the male. Okay, so that I mean you can see that it's the female is very fat in here. The fasted 1 month fasting is going to change this change. Cavefish, you do not see so much changes on the like body depth. I mean, this fish is still really having the big depths on the body depths, right? Tinaha is a new lineage. Morino is closest to the surface fish. Patron is further derived the population. The Tinaha is also a kind of known population, but they are not so much well spun, so we just use adult. Okay. All right. The gut structure, the surface fish is a little bit shrink and then it's getting narrower. If you actually dissecting this gut, it's very, very tender in the fasted condition. They're really thin and fragile. You can cut very easily versus this normal diet one. It's very stiff and then it's a very nice thick wall on the gut. All right. Okay, so this is this is where the Tinaha cavefish is. It's close to the Tachon cavefish. All right. So we did the adult. So the growth is not so much growing. We have a star, but you don't see that so much of the growth. Like effective size is small. The pre-fasting and post-fasting is control and the fasted individual. And we can see more effect on the body weight. Of course, they lose weight on the fasted condition. And the liver weight is more pronounced on the effect size is much larger than just looking at the body weight. So we take focusing more on the liver weight for this analysis. Okay. Interesting things is the Morino doesn't respond to the fasting. They don't lose the body weight. We don't know what they are doing, but they are dying. So that's a crazy population. So fasting, you would see the fasted individual have more distinct gut microbiota, and then beta diversity also showing okay, yeah, that is different, more separation from the normal diet feeding. We didn't see this one in the antibiotics, but the fasting have a large effect. And then these pathway analysis. Okay, so the liver weight. Okay, the important point we pay attention is the positive value here. Positive value is going to mean the different. So the fish are going to reduce the liver weight or the lower the body weight. Okay, so positive association means this pathway is also going to reduce. Okay, so the effect is going to be reversed. So if you see the positive association, this pathway is going to reduce, right? And then negative value here, this pathway is going to increase while the liver size is going to be smaller. Okay, they're losing weight, but this pathway is going on. Okay, that's there too. Okay, so the color code slightly changed, sorry, over the little bit mess. This orange is a Tinaha cavefish. Red is a Pachon cavefish. Okay, it's a Pachon, further derived one. Tinaha is it's similar but a distinct population. Surface fish in here. Okay, so the fasting response, it's the Pachon and the surface fish seems similar way than the Tinaha. Tinaha is really distinct. Gut length, again, gut length is shorter in the starved condition, and this pathway is the reduced in the Tinaha cavefish. There's a shared with the Pachon here, shared with the Pachon. But the point is, Tinaha is rather doing much more active change in the body, as well as the active association change with gut microbiota while they are fasting. So this is again, we did the meta-seq term analysis, and then this is all the cell structure biosynthesis, nucleotide biosynthesis. This, if you taking a look at these are more like the microorganism activity here. So microbiology, microorganism activity in the fasted Tinaha is shut it off. Okay, some mechanism Tinaha cavefish have this, while they are reducing the gut size or the body size, they seems to have reduced microorganism activity too. The reduced microorganism activity. We do not know how they are doing, but they do not have it. But instead, the surface in Pachon, they try to confront with this difficult condition by having the gut microbiota, which is have some carbohydrate biosynthesis. So some activity is booted up in the fasted condition in this Pachon surface fish. Please note that Pachon is okay for this fasting, but the surface fish is really suffered this 1 month of the fasting. They try to eat each other, and then their scale is really not unhealthy state. Some scale is going out not only for the physical attack, but their health condition is off, so their scale is also the bad shape. The gut length is a similar condition. I just emphasize fasting. This blue one is enriched with Tinaha. Okay, and the other rectangle, this green one is fish growth. So the positive meaning is the reverse, right? The growing fish have more on this system, but surface. This is surface fish here. So all in all, this the complex data set, if I plot into the 2D with the NMDS, the Pachon diet experiment, which means they are growing, okay, is in here. While when it is applied with the antibiotics, so they lose some association with gut microbiota, they move towards this way. So the enriched pathway is changing towards here. Surface fish diet experiment, when they are growing kind of close to this Pachon cavefish here, and that is going to the after the antibiotics treatment, they're going to here. Morino is in here. Again, this is a growth experiment, and we couldn't have the Tinaha the juvenile, so we don't have the Tinaha plotting here. Cell structure biosynthesis, which means the microbes enrichment in here. So when they are growing, they want to have some more microbes, which is have the actively dividing in their gut. The fermentation process abscess in here too. So that is helping their growth. But Morino is needing something different than these two populations. The F1 is actually they are really disrupted with gut microbiota. They don't have the positive association so much on the gut microbiome. Okay, liver weight, Tinaha, Pachon, and the surface, they are scattered very distinct position. Okay, we can understand that it's because they are different population. But so some the cell structure biosynthesis, which is for the microorganism, the proliferation, that is negative on this above axis, but surface fish they doing something on the gut. The microbes are doing something on the gut, fasted gut. Some vitamins, biosynthesis also bringing this surface fish in this axis. I told you that the tinaha is quiet for the many of the microbiome, microorganism activity, but somehow the fermentation is still on. Then pattern is on this here. So this complex data set, what we can pull out the conclusion is so cavefish, they adapt to the fasted condition, but they're very distinct in the terms of the gut microbiota. The tinaha have a more drastic change in gut associated pathway and the fasting. Tinaha seems shutting down the majority of the microbiota's activity, while some biosynthesis is still activated in the surface fish pattern. Then cavefish adapted a variety of way for the fasting, which we didn't expected for our analysis. All right. Okay, so that is all. So that is all the very wonderful team that's composed. Many of them is undergrad student. They're in the Europe project, and they're really engaged in the project. I put their name on the slide, each of the slide. But these are our team, and then our team is usually 10 or more undergrad student helping this project a lot. So we really appreciate the help. Thank you so much for your listening.